Hi, everybody, and welcome back to Geology 309, bringing fossils to life. This week we are going to be focusing on phylum Echinodermata, or the spiny-skinned animals. Really one of the more bizarre groups of life on the planet. So, the cladogram for Echinodermata shows a considerable amount of diversification over the Phanerozoic eon. And we have these three main groups, Crinozoa, Astrozoa, and Echinozoa, with subgroups within. We're going to go through all of these and look at some of the main features we see in all of our echinoderms, and also a little bit of some common taphonomic influences that we see on them as well. So we have 5 living classes of our spiny-skinned echinoderms, with lots of extinct classes, approximately 20. These have been around possibly from the latest Precambrian, definitely the early Cambrian to today. So they really do span the entire Phanerozoic eon. And while we've already talked about arthropods and mollusks, these are the third most common fossil phylum. So, if we look through which ones we still have alive today, we no longer have our eocrinoids, our cystoids, or our blastoids, but sea lilies, feather stars, starfish, brittle stars, sea urchins, and sea cucumbers, all still alive today with lineages that go back quite some time. All echinoderms are exclusively marine. Unlike arthropods and mollusks, are other abundant groups that we've discussed that could be terrestrial or freshwater or marine, all echinoderms are marine. And they're present in just about all benthic marine habitats. So from the deepest portions of the abyssal plain to the intertidal zone of the beach where you might vacation. However, relatively few are on shallow continental shelves. So while we do have a number there, it's not nearly as much as what we see in our deeper portions of the ocean. A lot of these can dominate tide pools. The ones we do find in our shallow environments are things like sea urchins and sea cucumbers, which can be living within the intertidal zone. Once you get below the photic zone, maybe couple meters below the surface of the water, echinoderms start to litter the seafloor in really great expanses. If you go down to the abyssal plain, 90% of the benthic biomass that you're going to see is actually echinoderms. It's brittle stars and sea stars. So we have lots of echinoderms in our deeper parts of the oceans, and they make up 90% of the benthic biomass. And the dominance that we see in the fossil record of shallow seas is really largely limited to the upper Paleozoic. So we have a brief period of time where echinoderms are really abundant in those shallow seas, and that's in the uppermost Paleozoic strata. But we don't see that traditionally throughout the Mesozoic, Cenozoic, or earlier in the Paleozoic. So, what about echinoderm ecology? All echinoderm groups start out with planktonic larvae and then a benthic adult stage. So because they have these heavy calcium carbonate plates making up their bodies, and we'll get into more of that in a moment, they don't freely swim very much. But as larvae, they're planktonic, which is how they become dispersed. So it's a wide dispersal during the larval stage, and then as they grow up, they settle into a benthic stage. They may be attached, sessile or vagrant, so we have some that stay put and some that get up and move around. And a wide variety of feeding strategies. We do have our filter and suspension feeding with things like crinoids, some detrital feeding, where they're going to be eating any organic material that's laying around. This would be our irregular echinoids. We have algal grazing echinoderms. These would be our regular echinoids, and some are actually active predators, such as sea stars. So again, wide variety of different feeding strategies within echinoderms. Yet few of them are very good swimmers. Again, their skeletons are made out of calcium carbonate. Actually, they are made out of calcite, but don't really have a buoyancy mechanism. So it's kind of hard for a sea star or a crinoid or a sea cucumber or an urchin to actively swim. Morphology of echinoderms is, well, frankly, bizarre. The skeleton is referred to as a test, similar to what we use with microfossils, and each test or skeleton is composed of individual plates or ossicles of a single high magnesium calcite crystal. So each plate on a sea star, on a brittle star, on a sea cucumber, on a sea urchin, or every spine on a sea urchin, or every plate on a crinoid is an individually grown high magnesium calcite crystal. So these tests are then encased in an organic skin, so their skeleton is essentially an endoskeleton. Even though that skeleton encases a coelom, they are still a coelomate, they have this organic skin that covers the test, producing one of our first invertebrates that actually has an endoskeleton. Finally, they grow by ossicle accretion and addition throughout ontogeny. So they just add plates or add onto plates, but their growth is very different than what we might see with arthropods or mollusks or, well, even vertebrates. Echinoderms generally have 5 ambulacral areas. So we'll see in a moment what those look like. These are 5 regions that indicate some kind of pentameral 5-fold symmetry. So think sea star, think blastoid, think crinoid, and all of them have these 5-fold symmetry. Those actually extend out into what we call ambulacral areas. Sometimes, such as with our irregular echinoids, we actually have bilateral symmetry, but there are still 5 ambulacral areas. Larvae are always bilateral, so when they're planktonically floating as larvae, they have bilateral symmetry, and then depending on what group they are, when they start to grow, they will either develop continued bilateral symmetry or they will branch out into 5-fold pentameral symmetry. And one of the most bizarre things about echinoderms is their vascular system. They don't really have a vascular system with blood the way that we would think about it today. All of their movement and everything with their physiology is done through a water vascular system. Essentially, it's a hydraulic pump. So when a echinoderm, like a sea star, for example, wants to walk across the seafloor, they use hundreds of little tube feet on the bottom of their bodies that are activated and moved by pumping water from one part of the body to the other. And so it acts as a hydraulic system, and that's how they actually move around. They regulate the osmotic pressure of this hydraulic system by pulling in seawater through a little valve on a porous plate that we call a Madreporite, that they can essentially change the hydraulic fluid, which is just seawater. So their nutrients are moved around through seawater and all of these things. Everything is done through hydraulics. They're kind of steampunk animals. And so they will squeeze that water like hydraulic fluid from one tube to the next. So their muscles are essentially just tubes or a vascular system that can constrict or relax, and that moves seawater through tube feet, extending into the ambulacral areas. So all of their movement is done this way. They really only have a sense of touch. They don't have a sense of sight. They don't have a sense of hearing. They don't have a sense of smell or of taste, but they do have a sense of touch. And they don't have a true circulatory system. They don't have a true respiratory system or excretory system. It's all hydraulics. Literally, steampunk animals. Yet, they are deuterostomes, not proterostomes. And if you remember the difference, it's during that first blastulation of cellular mitosis, what that first gastrulation forms. Is it the anus or the mouth? In this case, they're deuterostomes. So it's anus first, and these are why they're also more closely related to hemichordata and chordata than any of the other groups we've looked at. So out of all of the phyla that we've discussed up to this point, echinoderms are more closely related to us than they are other invertebrates. Let's look at our 5 living classes of echinoderms. First, we have class Asteroidea. These include our sea stars, or so-called starfish, and these have a fairly minor fossil record. Class Ophiuroidea are brittle stars. So brittle stars and sea stars are not the same thing. These have a very poor fossil record. You usually find them more in Lagerstätten style deposits. Class Holothuroidea includes sea cucumbers, very poor fossil record, very strange group of animals that do some weird defense mechanisms, such as extruding their digestive, or their guts essentially, out as a way of deterring predators. Class Echinoidea includes our sea urchins and sand dollars, and these have a fairly good fossil record. And then finally, our class of Crinoidea, which include crinoids, very good fossil record, but a minor group today. So what you notice here is that brittle stars, sea cucumbers, don't really have much of a fossil record, and that's not uncommon really among all echinoderms in general. Because their bodies are composed of that calcium carbonate test, all those the skeletal elements, and a thin organic layer over them, when they die and decompose, those plates tend to disaggregate and separate. So brittle stars, sea cucumbers, their parts just kind of fall apart and get cast around, and they will redissolve into seawater, especially considering that most of these are going to be living in deeper portions of the oceans. However, when we look at crinoids and some of our echinoids, these are more shallow water, and we're much more abundant, especially the crinoids. So our crinoid fossil record is very good, but it's predominantly lower Paleozoic. So here's class Asteroidea. Again, Ordovician to today, but kind of a mediocre fossil record. So their mouth is ventral, so the mouth is on the downward surface that would be close to the sediment, and the anus is above on the outside. The ossicles that make up their skeletons are very, very small, which again, why when they die and that organic layer dissolves, these ossicles are going to just cast out. They are active predators that have very powerful tube feet. In the upper right corner, you can see a close-up of some of those suction cup-like tube feet, and they use these to literally pry open bivalves. So these are predatory, and they're very, very strong, especially for their size. You can see that Madreporite on the dorsal side, on the back side. It's sometimes a slightly different color, but it's off center a little bit. So, for example, if we get my highlighter here. So this is the Madreporite, and on the arboreal, or I should say dorsal side, you can see that here is the anus, and then the Madreporite is that valve right next to it. On the ventral surface, or what we call the oral surface, the mouth is in the middle here. And then these are the ambulacral areas and the ambulacral grooves that we were talking about, these 5 regions on the underside of the body. And that's where the tube feet are going to be, and they all radiate towards the mouth. Class Ophiuroidea includes our brittle stars. Again, they look a little different. They're not as robust. These are actually a modified filter feeder. So brittle stars they're not predatory. They essentially filter feed. And they just kind of hang out. You can get these for aquariums. On the ventral surface, though, their anatomy is not entirely different. They do have a mouth in the middle on the ventral surface. But instead of having the tube feet, they just have a lot of ventral plates because they're not trying to pry anything open. Class Holothuroidea are the sea cucumbers. Again, a very strange group. There is no skeleton other than some internal calcitic spicules, almost sponge-like. Here you can see one that is doing the gut extrusion as a way of detracting and deterring any kind of predator. So they have kind of a mouth on one end and the anus on the other. So essentially food goes in this way and comes out that way. But the internal structure are a series of the oddly shaped spicules that you can see diagrammed before you. Class Echinoidea. In this case, the calcite plates fuse together, which gives us a slightly better potential for a fossil record. The mouth is usually at the bottom. These are unattached. They can be epifaunal or burrowing. Some burrow very deep, some burrow very shallow, and some live right up at the surface like the spiny urchins that you can see. They still, even the spiny urchins though, do still have tube feet. So you can see these strands of tube feet coming off and then the actual spines themselves. So they will use the spines to help move, but they also have these long tube feet that'll help them pull around. So their motility is, and feeding is a mixture of the movement of both of those. We have two subclasses of Echinoidea. We have regular and irregular echinoids, and a lot of this depends on whether it's a spiny urchin that is perfectly pentamerally symmetrical or a more flattened irregular urchin which has less spines. So these would be our regular echinoids. As you can see, they're spiny urchins. Sometimes the spines don't preserve well, but we're left with these little nubs where the spines were anchored on in kind of a ball and socket structure. You can see the tube feet coming off of here much better. So subclass Regularia. Again, notice the fusion of these plates, but as an echinoderm, each plate is its own high magnesium calcium carbonate crystal. Each spine on the spiny urchin is a high magnesium calcium carbonate crystal. So these are so-called sea urchins. We have very primitive forms in the Ordovician, but they're overall pretty uncommon in the Paleozoic. We start seeing these more in the Mesozoic onward. Some of them are very spinos with large single crystal spines for protection and for movement. And actually, these are epifaunal grazers. So if you're walking along the surf of the beach and you step on a spiny urchin, it's going to hurt you. It's going to hurt it. And it wasn't trying to hurt you, though. It was just an epifaunal grazer. They mostly eat algae. However, they have this radial symmetry. You can see the 5 ambulacral areas, but even the inter areas are also showing pentameral symmetry. However, they don't really have a front or back. So a regular echinoid can't really move forward or back. They just move in a direction. There's no head direction or tail direction. The mouth is on the bottom, anus on the top, as we've seen with other echinoderms. But this is a really kind of an interesting group. Who knew that a ball of spines could actually be an animal? And hydraulic. If you look inside of an echinoid, though, you can see the mouth structure here made of, again, a series of calcium carbonate plates. Now, what about our irregular echinoids? These include our heart urchins and our sand dollars. These are known from the Jurassic to today. Again, fused plates. These show bilateral symmetry. Now, they still have the 5 ambulacral areas, but they have only bilateral symmetry. Only one way you can cut them to make a mirror-like image. These are infaunal for the most part and do have directional movement. So there is a front and a back. Some of them have hair-like spines, especially the heart urchins, but they're not nearly as spinos and rigid as what we see with our regular echinoids. In this case, also, the mouth and anus are both at the bottom, and that's probably part of it being that they're infaunal. As a result, there's no surface that they can just excrete out of, so they have to have the mouth and anus separated from each other, but both on the ventral surface. Heart urchins are around from the Jurassic to today, and because they're a little bit thicker, they're actually a deeper infaunal echinoid. And our sand dollars, which have been around since the Miocene from today, these are the flatter ones, and these are a shallow infaunal. So just like we looked at with bivalves, the morphology can tell you about lifestyle. So the thicker and more kind of swollen looking the echinoid is, the deeper it probably burrowed for irregular echinoids. And shallow sand dollars are a shallow infaunal organism. We do have some very interesting trace fossils around the world from various types of echinoids. So very similar to what we see today with how sand dollars and urchins move around. We also see records of that in different types of facies from the Paleozoic, Mesozoic, and onward. And we're going to get more into trace fossils in an upcoming lecture. Now, what about our extinct classes? Well, we have up to 20 extinct classes. All of them are Paleozoic. One of the more interesting ones is class Blastoidea, including our blastoids. Blastoids appear in the Silurian and go extinct in the late Permian, right at the Permo-Triassic extinction, yet their acme event, when they are most abundant, is in the Carboniferous, especially in the Mississippian. These are part of that Carboniferous reef complex where when corals are at an all-time low. They have this little bud-like calyx or head at the top. The calyx is the main portion body of some of these echinoderms. In fact, the nickname for blastoids is the sea bud. They don't have any arms, but they did have likely very powerful tube feet. They were attached but not strongly stalked, and this is to differentiate them from the more diverse group, the crinoids. So usually when you find a blastoid fossil, you're just finding that calyx. But again, note the ambulacral areas and the pentameral symmetry on the top. So the mouth is in the middle, and these ridges, you can see the tiny little ridges almost like the edge of a dime going down the ambulacral area would have had little fibers on it and tube feet that would have helped move food up the ambulacral area and down into the mouth. Class Rhombiphora includes the cystoids. Now, cystoids are weird. It's a polyphyletic group. We see these in the lower Ordovician through the middle Devonian, with their abundance being in the Silurian, but even then, they're quite rare. Cystoids are strange because they have a very irregular shape. It's lots of calcium carbonate plates, but somewhat ovoid or globularly attached to one another. They may have been unstalked or had very small stalks. The cystoids are kind of the black sheep of the echinoderms. We also have a very, very strange group that's also probably polyphyletic, Stylophora or the carpoids. These are just from the Middle Cambrian to the Middle Devonian. Again, asymmetrical, though they probably could have moved around more than cystoids. Not a lot is really known about this group. Again, probably polyphyletic. The class Eocrinoidea, though, from the Cambrian through the Silurian, is an early branch of what we're going to see with other crinoids. These have a poorly formed calyx, but lots of branching arms and short stalks. But this kind of leads us into crinoids. So let's take a look at class Crinoidea, which is where most of our echinoderm fossil record is contained. Crinoids make up a significant portion of our Mississippian bioherms. Since most of our corals are gone by the Mississippian, these are the basic framework of reef type and bioherm complexes. They're also important for the production of a higher level tiering going on both infaunal and epifaunal in the Paleozoic. So as a result, a lot of the Mississippian limestones are fossiliferous limestones made almost exclusively of echinoderm parts. Class Crinoidea includes a stem or a stalk that's produced of these little kind of Cheerio or poker chip shaped columnals, these little rings that are stacked up and which also branched out and formed little tentacles or cirri branches. The stem will terminate into a holdfast, and it looks kind of like roots, but these are actually holdfast tube feet, and that's going to hold them into the sediment. Now, however, crinoids were capable of unrooting themselves, walking across the seafloor, and reattaching somewhere else when they wanted to. So this would be almost like watching a tree unroot itself and walk across your yard. The main body, again, is formed of a calyx, not entirely dissimilar from blastoids, but more cup-shaped. Right at the edge or lip of that cup-shaped calyx would have been 5 branching arms, each one an ambulacral area. And these are also often subdivided into smaller branches yet. Each arm has an ambulacrum on the upper side, and this outpoaching of that proper coelom internally. In other words, they use these as part of their feeding. They also would have been lined with little calcium carbonate pinnules to help with filtration and moving food particles down the ambulacral groove with tube feet towards the mouth. So they're doing something very similar to what blastoids were doing, but they're branching out with these needle-like arms, moving food and kind of shoving it hand over fist into the mouth of the crinoid itself. So the top of that oral surface is known as a tegmen, and it's the mouth, and then the very center of it with the anus right off to the side. And we also see this with some of our embryoasteroids, and we see it with blastoids as well. So the mouth, the food goes in one end, and it kind of gets kicked out, waste gets kicked out of the side. The arboreal surface is actually down towards the stem. So they have the oral surface facing upward. And crinoids don't move much because they probably were able to survive off of a relatively low amount of nutrition. Remember, the more you move, the more calories you burn. So if you just stand in one spot and you can filter feed or suspension feed, you may not be getting much food, but you're not really doing much, so it's fine. They could probably crawl. We know that some could swim. We know that some of our modern crinoids can swim, but not super well. But we see that once they attach themselves somewhere, as long as the feeding is good, they don't really do much. They just kind of stay there and absorb. So here are some diagrams and more schematics of our echinoderms. Again, we have the stem produced of individual columnals. The calyx is here, and you can see there's various calcium carbonate plates. Those become very important whether we are looking at a radial plate or a basal plate or an infrabasal plate. The pattern of that is used for phylogeny. Here's the arms branching off with the individual pinnules, and there's that ambulacral groove right down in the middle. So the mouth is in that tegmen, right in the middle of the calyx. Okay, so they're going to branch out their arms, they're going to collect food, and then those pinnules will help move the food down the ambulacral groove right towards the mouth where it can digest. Again, we use the calyx plates as a way of identifying different types of crinoids. You can't always just go off of a columnal or stem piece. So at a minimum, a crinoid calyx has two different rows of plates. The radial plates, which occur directly below the base of the arm, so right along, making up that lip right at the edge of the calyx. The basals are just below the radials and alternate with them, so it's almost a checkerboard-like pattern. So if we look at these two different types of crinoids, which we'll get into monocyclic and dicyclic here in a moment, but in pink we have our radial plates, and in yellow we have our basal plates. Notice the radial plates are the ones that directly attach to the arms, and the basal plates are immediately underneath our pink radial plates, and they alternate in their placement. Now, some crinoids, what we call dicyclic crinoids, have infrabasal plates that are underneath the basal plates right before it gets to the stem. If you have a crinoid that lacks infrabasal plates, we call them monocyclic, and if they have infrabasals, we call them dicyclic. So, and I just realized I spelled infrabasals wrong. So if you want to figure out if your crinoid is monocyclic or dicyclic, you start at the arms and then work your way down onto the calyx and identify the radial plates. Then look below that and find the basal plates. Once you've identified the basal plates, look beyond that. Is it going immediately to a stem? Then it's monocyclic. If there's another set of alternating plates underneath the basals, then those are infrabasals, and those would be dicyclic. So we can explode view some of our monocyclic and dicyclic calyces and see that we have, in case of this example up here for a monocyclic, we have our arms, and then the black ones are going to be our radials, and inside are our basals, and that's it. Then we go into the stem, that's monocyclic. In the case of a dicyclic, we have our black annotated radial plates. We then have our basal plates alternating past that, and then alternating yet again the infrabasals and then the stem. We have three major groups of crinoids. We have all of them starting in the Ordovician and then either going extinct at the Permian or still living today. So obviously the Permo-Triassic extinction was a major hit point for crinoids. Our three major groups are Disparida, the Camarids, and the Cladids. So Disparida, Ordovician to Permian, the Camarids, Ordovician to Permian, and Cladida is Ordovician to today. And there's two groups within Cladida, the flexibles and the articulates. The flexibles went extinct at the end of the Permian, and the articulates from the Carboniferous and actually still around today. So here's a generalized phylogenetic tree of class Crinoidea. Notice our rhombiferous cystoids are kind of the sister group and the outgroup here. We have subclass Disparida, and then Camarids and Cladids are more closely together. So let's start with subclass Disparida. Again, Ordovician through Permian. These are all monocyclic, so they're all going to be lacking those infrabasal lower plates before you get to the stem. These have a very simple structure of the calyx where it's radials, basals, what we call anal plates, which is part of the kind of excretory system, and then some forms of brachial branching plates. Their arms are very simple and lack pinnules, and they contain very small forms. The calyx's heights here are only about 1 millimeter tall, so disparate crinoids are very, very, very simple. And this is essentially what they look like. You've got the arms that come down, and they just kind of morph into a calyx, which morphs into a stem. Very, very simple forms of crinoids. Class Camerata, again, Ordovician through the Permian. These are a little bit more complex. As you can see from the fossil calyces illustrated here, there are more plates, and there's definitely a differentiation from arms to calyx. It's not like the Disparids where it just kind of morphs from one to the other. So if you actually do an exploded view of these calyces, they're much more complicated. Their calical plates of the calyces are rigid, so they were really locked in place. The mouth plate area, the tegmen, is very, very thick, so the plates there are really, really thickened. And they have a fixed brachial and interbrachial plates that form part of the calyx. So again, the arms are really well defined in this case. Also, the arms are biserial, and they always have pinnules. So the arms branch into sub-branches, and all of those have pinnules on them. And then subclass Cladida, again, Ordovician to recent. These are all dicyclic, and they're subdivided into our two clades, the Flexibilia and Articulata. So let's take a look at those. Subclass Flexibilia from the Ordovician through the Permian. The calyx and the tegmen here are flexible, and they have open food groups on the tegmen, so they are able to be kind of moved around a little bit more. They have a little bit of some similarities to the Disparids. The arms are distinctly differentiated from the calyx, however, so you can definitely see when the arms branch off. Brachial plates are not part of, joined to those of the calyx with really, really strong articulations. So those plates that make up the base of the arms aren't super well attached. The arms also lack pinnules. Again, these can easily be mistaken for disparate crinoids. And they also don't have any cirri, little sub-branches off of the stem. And subclass Articulata, these are the ones from the Carboniferous to today. These are the ones we still have alive today. These are also dicyclic. Their infrabasals are very, very small, and some of these have completely lost their stalks altogether. The arms are very distinctly differentiated from the calyx. Brachial plates are not joined with any strong attachments of the calyx either. And as you can see from some of our modern images here, yeah, they still have a holdfast, they still have a calyx, they have arms, but they've lost, some of them have lost their stems. The arms are uniserial, so they just kind of branch out. They don't sub-branch, but they have lots of pinnules on them. All post-Paleozoic crinoids are subclass Articulata. This includes a lot of our recent diversifications of the stemless Comatulidas. So these are the ones that actually can swim. So I recommend you find some YouTube videos, look up modern crinoids, and you can see these things will pulse through the water by opening and closing those arms. They'll actually, almost like a balloon, will float through the water. So what about our crinoid ecology? In the Paleozoic, from the Ordovician through the Mississippian, crinoids essentially are very common in shallow water facies. We see a lot of them in our back reef facies and on the landward side of barrier islands. So they're really taking up a lot of ecospace in these shallow water environments. These are also places that have well circulated water, but there's not too much energy. As you can imagine, crinoids wouldn't do too well with a lot of storms and high wave action. So there's movement of the water, enough to keep sediment kind of out of the way, but it's not so strong that it's going to do damage to them. After the Permian extinction, our surviving crinoids are quite rare, and our modern crinoids are restricted to our deeper parts of the ocean, where we're looking at over 100 meters of depth, with one exception. We have one shallow water crinoid today. So here you can see an image from the abyssal plain of our ocean floor. And again, we have crinoids, and you can even see some starfish there too. Again, over 90% of the biomass that we have making up our abyssal plains are echinoderms. The Comatulids are among our most diverse living group, and these are the ones, the shallow water stemless crinoids that we see today are very common in our modern reefs. And these can get up and walk around on their arms, or they can swim, but they don't use stems. And again, they're suspension feeders, so they'll get up and move around and find a place where there's lots of organic material in the sediment or in the water column that they can filter out from suspension feeding. After the post-Paleozoic has been a really rough time for crinoids. They've either retreated to areas where there's no predators like the oceans, or they've evolved the ability to get away from them faster like the Comatulids. So it's interesting. This is a good opportunity to really look at how ecosystems change. So when we look at our Paleozoic echinoderms, they're in shallow water facies. When we look at where we find those same groups today, it's in deep water facies. And so there's an evolution there in ecology, and there's reasons for it, though. Again, avoiding predators, either by getting away from them, or by living away from them, or by being able to quickly get away from them, are two really interesting changes that echinoderms, like crinoids, have taken. So here you can see some better views of some of our modern day crinoids compared to some of our Paleozoic ones. So both of these, in this case, are shallow water crinoids, but they're structured very, very differently for very different reasons. So that's it for this week's look at echinoderms. Next week, we will have a short lecture on ichnofossils and plants. And then after the break, we will be rounding up the semester by looking at chordates. So have a good week.